Chapter 7 looks at the cellular basis of inheritance. Multicellular organisms, those that live as masses of communicating cells or tissues, all begin as a single cell with genetic information organized through a process known as meiosis. We're going to look at sexual reproduction in this chapter with a focus on the creation of sex cells, known as gametes. The birthing of offspring that look different from the parent is an evolutionary adaptation that's developed over millions of years. Maybe those offspring that look different might obtain an advantage from a novel mix of genes thanks to the life cycle of their parents. When we talk about life cycles in biology, there are three major ones that you can consider. The first is the diploid dominant life cycle. In diploid dominant, the multicellular diploid stage is the most obvious life stage. There is no multicellular haploid stage. That means that for the vast majority of the life of a diploid dominant organism, all of their cells, except for their own sex cells, are 2N. At no point in time in their life do they have any 1N cells that are more than just a single 1N cell. In the case of humans, either a single egg or a single sperm. In haploid dominant life cycles, there are multicellular organisms where every cell is haploid. In fact, in the most obvious stage of their life cycle, all of their cells are haploid. You see this with all fungi and a few different types of algae. They do have certain points in their life where they exist as diploid organisms, but that's when they're preparing to reproduce to make more haploid organisms. The third life cycle that we can study is the alternation of generations, in which two stages, both haploid and diploid, are apparent to one degree or another, depending on the point in the life cycle that you're studying. You see this in a lot of plants and some other types of algae. We'll talk about these second two life cycle types when we study those groups more closely later on. Today, we're going to focus on the creation of sex cells or gametes in the diploid dominant life cycle individuals. The major assumption in this particular chapter is that you have a very strong understanding of the steps of mitosis and what exactly occurs in each of them. So if you're shaky on mitosis, I'm going to tell you to stop right here and study mitosis, or at least get your notes in front of you. Because the process of meiosis is essentially mitosis times 2. You run through all of the steps, but you skip interphase when you run through meiosis for its second go-around. We break up the stages of meiosis into meiosis 1 and meiosis 2, which makes it a whole lot easier. This is what I mean if you're having a hard time picturing it. Let's wrap our heads around the process of meiosis first, and then we'll consider what makes it unique. Let's start on the bottom of this figure. You see the regular steps of mitosis. You begin in interphase, where you duplicate all of your genetic material, head into prophase, where you organize it, prometaphase, where you start to attach your spindle fibers to your chromosomes, metaphase, where you line them up, anaphase, where you pull them apart, telophase, where you organize them into 2 new cells, and then finally cytokinesis, when you actually have those 2 new cells. Up top in meiosis, you'll notice those exact same stages are repeated twice. But when you go to repeat them the second time, you skip interphase. So let's walk through the process. In meiosis, prior to the division process, you go through interphase. You duplicate all of your chromosomes. You line them up in prophase 1, and we'll learn about a special step that happens here called the crossing over. But let's just leave it at in prophase 1, crossing over occurs. In prometaphase, you attach the spindle fibers to your individual chromosomes. In metaphase, you line them up, and in anaphase, you pull them apart. In telophase, you end up with 2 cells that have 46 chromosomes yet again, if you're looking at a human being. Now, we head into meiosis 2, and this is where we hit prophase, metaphase, and telophase 2. All labeled, as you can see, with that little Roman numeral to let you know we're looking at the second division. In prophase again, we organize our chromosomes. In prometaphase, we attach spindle fibers to them. In metaphase, we line them up. In anaphase, we pull apart individual chromosomes into chromatids, and what we make are 4 cells that are now 1N. We started with a 2N cell, we went through interphase to essentially make it 4N, and we split it into 2 2N cells. With that second division, skipping interphase, we end up with 4 1N cells. So meiosis takes one normal cell and gives you 4 potential gametes. And thanks to the special events like crossing over, you end up with 4 potential gametes that all look really different from one another. That's why while you might share some general physical characteristics with your siblings, unless you're an identical twin, you don't look exactly alike. So let's learn about what makes meiosis unique. The anatomy of chromosomes needs to be explored before we get to those points, because if we're not quite sure what all the terminology is, it's very easy to get lost. These 2 pictures are taken from the internet. They're not found in your textbook. So let's just look at exactly what we're talking about before we head into all of these neat features so we can make sure we're all on the same page. In one single duplicated chromosome, you have 2 sister chromatids. They contain the same information, as in they both contain copies of genes, 1 through 37, but maybe they don't contain the exact same copy of the gene, but they all have genes 1 through 37. In the middle of the gene, you have a centromere. It's a special protein complex that's holding those chromosomes to one another. The ends of the chromosomes are known as telomeres, and when you split the chromosomes apart or look very closely at that centromeric waist, you'll notice that they have individual kinetochores. That's the handle that the spindle fiber is connecting to. Here's the part that really throws off a lot of students. When you have a chromosome that's X-shaped, we call that a chromosome. But when you split it in half and you only have half of the X, we still call that a chromosome. So it can get a little messy. It's important to note if you're talking about a whole chromosome as in an entire X or just one of the sister chromatids. I'll do my best to always point that out to you. I know that it's confusing, and if there's one thing in biology that I could change, at least when it comes to genetics, this would be it. So consider the anatomy of your chromosomes, maybe doodle it on your notes, and we'll move into what these little guys are capable of doing. The first major kind of cool effect of meiosis is crossing over. In this picture, we're just going to consider 2 chromosomes that are identical. The blue chromosome came from this person's dad, and the red chromosome came from their mom. You can see that they are homologous chromosomes. These are both chromosome 1s, and they both carry genes for the product A, B, and C. They just so happen to carry different types. That's completely okay. You can have different alleles for genes. So what happens in the process of meiosis, specifically prophase 1, you have crossing over occurring between non-sister chromatids of homologous chromosomes. These chromosomes are homologous because they're both chromosomes number 1. However, their chromatids are not considered sister chromatids. They contain different pieces of information. So if we cross over pieces of non-sister chromatids, you end up with recombinant chromosomes as seen below. Now you have a little bit of a mix in your chromosome pair. Both chromosomes have everything that they need. They both have copies of A, B, and C, but they've been reorganized. Chromosomes that have a mixture of maternal and paternal sequences are referred to as recombinant chromosomes. And this will mean that the child that results from these gametes will have a unique mix of characteristics and therefore look a little bit different than mom and dad. Another factor that helps with the individuality that comes out of meiosis is random independent assortment. Let's look at a very simple sample. This example occurs in metaphase 1, and for this individual, they only have 2 chromosomes. You can see that in metaphase, these chromosomes are lined up along the metaphase plate. Their microtubules, those spindle fibers, are attached to their kinetochores, and they pull apart the chromosomes just like normal. In this instance, they randomly align where one gamete got 2 blues and one gamete got 2 reds. Well, there isn't any law dictating that. As an example 2, you can see one gamete got a red and a blue and one gamete got a red and a blue. And when you split them up again randomly, you find that you have a lot of different genetic arrangements that are possible. This random independent assortment of chromosomes allows for more individuality and more variability between our individual gametes. When there are more chromosomes, the possible number of chromosome arrangements increases dramatically. This was a law found by a man named Gregor Mendel. We'll talk about him in just a little bit when we get to genetics. Let's look a little bit more at the movement that occurs in the chromosomes, or among the chromosomes, between the different steps of meiosis. Remember, all of the steps of meiosis are exactly the same as the steps in mitosis when it comes to what's happening in them and the order in which they follow. There's just a little bit extra in meiosis 1 and meiosis 2. For instance, meiosis 1 starts with interphase, G1, S, G2. Then you head into prophase where you organize your chromosomes and maybe they undergo a little bit of crossing over for rearrangement. Next, they head into prometaphase 1, where the microtubules attach to the fused kinetochores on the different homologous chromosomes. In anaphase 1, the homologous chromosomes are separated from one another. You head into telophase, like normal, and cytokinesis, like normal, but then you head straight back into prophase, which 2 cells that only have 46 chromosomes. So this is where it gets kind of neat. In prometaphase 2, the microtubules are going to attach to the individual kinetochores on sister chromatids. It's much different than what's occurring in prometaphase 1. So when you move into anaphase 2, after your prophase 2 and prometaphase 2 and metaphase 2, you see that your sister chromatids have been separated. So there's a whole lot of movement occurring in this process of meiosis. Let's compare the processes of meiosis and mitosis. You'll see that while they share a lot of the same steps, the variability in the endpoint comes from the different types of behaviors that the chromosomes undertake during the different stages. For instance, there's a synapsis of homologous chromosomes that occurs in meiosis, but not mitosis. The synapsis is this tight pairing of the chromosomes right next to one another. That occurs and it allows for crossing over. The second difference they bring up in this process. Crossing over occurs in meiosis, but obviously not mitosis. You don't want a reshuffling there. You also find that homologous chromosomes line up next to each other during metaphase 1 of meiosis, but not at all during mitosis. Those homologous chromosomes get pulled apart differently in meiosis so we can ensure we make our nice randomly assorted gametes. One thing that is shared between part of both of these processes is the lining up of sister chromatids at the metaphase plate. Sister chromatids line up at the metaphase plate during mitosis when we're looking at metaphase because we would like them to be pulled apart to make 2 identical cells. However, the sister chromatids that are lining up at metaphase 2 of meiosis have undergone crossing over and they look quite different from one another. Thanks to all of these strange little rearrangements and synapses and crossing over and lining up at different times, we find 4 haploid cells at the end of meiosis 2, while there are 2 diploid cells that are identical to one another at the end of mitosis. If you're curious what all of these chromosomes might look like and where we've gotten all this information from, welcome to a karyogram. A karyogram shows the chromosomes of an individual organism lined up. In this instance, they're lined up from 1 to 22, or tallest to smallest. The 23rd pair are the sex chromosomes. In this instance, you can see that this individual has 2 X chromosomes and is therefore female. We can actually diagnose some genetic illnesses by looking at karyograms. There are some illnesses that might be familiar to you even if you haven't thought about them in a karyographic kind of way before. For instance, if there's a 3rd chromosome 21, that individual would have trisomy 21, colloquially known as Down syndrome. Issues like that come about from errors called non-disjunction. So these are errors in the process. Following meiosis, each gamete should have one copy of each chromosome. However, in certain cases, the chromosomes are not pulled apart correctly in either meiosis 1 or meiosis 2. They simply fail to separate from their partner. So you end up with cells that are either missing a chromosome or they have an extra chromosome. We find that missing or having an extra sex chromosome is actually a lot less dangerous to the body than missing one of those chromosomes 1 through 22. Those are your somatic chromosomes that contain the genes that run the rest of your body. We find that these errors have a much higher incidence when the maternal age increases. As you can see in this graph here, the risk of Down syndrome in live birth by percentage shoots up drastically after the age of 40. This is why there's a lot of research surrounding the Down syndrome correlation and its ties to maternal age. Some fertility physicians would like to select for healthier eggs, while others simply wish to understand the mechanism behind their creation. Another common topic of research when it comes to chromosomes is X inactivation. I've mentioned previously that having an extra chromosome can be dangerous. Well, women have 2 X chromosomes and men only have 1 X and then a Y. Is it dangerous for women to have that second X? The answer is no. Second X's in cells are inactivated. They're essentially crumpled up into something called a bar body. B-A-R-R, bar body. Those bar bodies are not capable of being used for any type of genetic expression. So the cells aren't harmed in any way by having too many proteins or too many enzymes. A neat kind of visualization of this idea is a tortoise-shell cat. In this instance, you can see my cat, Annabella. In her body, every single cell has lost 1 X chromosome, and it just so happens that in cats, their fur color is coded for on their X chromosomes. Because one of her X's has been randomly shut off, some of her genes have been randomly shut off. You end up with cells that make her fur color with different X chromosomes. In some instances, there are black X's that have been left large and useful, and in other instances, there are orange X's that have been left large and useful. So a nice visualization for X inactivation is this coat color that you see on Annabella. Another common instance of study is, of course, what happens, again, when things go wrong. One of the more common things that go wrong is inversion. In the case of inversion, crossing over occurs, but the little splice that gets taken out of a chromosome ends up being flipped upside down in the chromosome that it lands in. Another example of an error is reciprocal translocation, and that means that the end of one chromosome is broken off, and instead of getting added to the end of its sister, it gets added to the end of a different chromosome. Both of these issues can cause major problems, or they can be considered silent mutations. That simply means mutations without major effects. We'll learn more about genetic information and genetic illnesses when we get into our specific study of genes. For that, though, it's the end of our chapter. This can be a very tough topic for some students, so give yourself some time. Read multiple resources, listen to a lot of different videos, and consider many different explanations. Work slowly, ask questions, and take a crack at your homework.